myself Dr. Pallavi Railahale, and I am going to teach you plant tissue culture today of BP 405T. Today we are going to start with the topic of types of culture. Before we move further to types of culture, I would like to mention three terminologies here, so that we have more understanding of these different types of cultures. So, the first term which I am going to use is the explant. Now, what is a explant? Explant is the part of the plant which we are going to use as a source for cells. Like if I have a plant, I like it based on its properties, like it is disease resistant, its yield is very good. Of yield means what? Of secondary metabolites. Its produce is of good quality, means if I talk about the leaves, there is lot of foliage in the plant. If I talk about the fruit, it is a good sized fruit. So, it is having all the properties which I want in my new plant. So, I will choose that plant and I will take a part of that plant for doing my plant tissue culture, and that is my explant. Okay. So, this is the explant. Now, once this explant is taken, the next thing which is done is that this explant is first of all you can say disinfected. Generally 10% sodium hypochlorite solution is taken. After that, again it is washed with water, which is sterile water, and after washing it is cleaned and put in a medium containing any kind of agar along with various nutrients. Okay. Now, this leaf which we had taken, we are going to cut a small section of that leaf and we will place those leaf sections in the petri dish. Gradually what will happen, that we will incubate it further. Incubation will be either generally at 25 degree centigrade with a relative humidity of 40%, and we will give a proper cycle of sunlight. Like in normal conditions, we have 12 hours of sunlight, 12 hours of no sunlight, okay, darkness. So, that cycle is there in the lab also. So, we will keep this petri dish in all these conditions at 25 degree centigrade, 40% relative humidity and 12-hour sunlight cycle, photo cycle. After that, after 7 days, what will happen, that these explant will get converted into undifferentiated mass of cells, which is known as the callus. It is known as the callus. Okay. So, now this callus is our main source for further development into newer plants. From this callus, we will take some part and we will transfer it into a fresh medium and provide plant growth regulators, so that this callus starts generating into a new plantlet. Once you have the new plantlet, you can further transfer it after some time into the soil and develop an entire plant from the plantlet. Okay. So, this is in short the process of plant tissue culture. Now, coming back to our different types of plant tissue culture, generally they are classified based on, first of all, the type of organ. So, it is known as the organ culture. Then further they can be specialized type like the hairy root culture, as well as protoplast culture and somatic hybridization. So, we will be reading about these topics later on. First of all, we will be learning about the organ culture, because they are more widely performed type of culture. So, what is organ culture? As the name suggests, which part of the plant we are using to perform the plant tissue culture, that is the organ culture. It is very simple. So, first of all, you have root tip culture, which is where you use the meristematic tissue of the roots. You have the shoot tip culture, where you use the meristematic tissue of the shoots. Then you have the leaf or the leaf primordia. Leaf means, you understand, the leaves of the plant, and leaf primordia is the area of the top of the leaf. If you see any kind of plant, on the top you will find this kind of a structure, and this part is known as the leaf primordia. It is again a rich source of meristematic tissue, it is like a shoot tip. Then you have the flower culture, where you use the flower parts for doing the culture. Anther culture or the pollen culture. You have ovule and the embryo culture. Ovary culture is performed. Nucellus culture is performed. Seed culture is performed. Endosperm culture as well as fruit culture. So, we have different types of culture depending upon which part we are going to use for doing the plant tissue culture. So, first we start with the root tip culture. Here first of all, the lateral roots, basically you have the in a tap root system, this is a tap root system where you have the lateral roots coming in like this. So, when you take any section of this area where the lateral root has originated, so we will take the lateral roots, that is one option, or we can take the tips of the lateral roots. So, we can take either the tips or the part where the differentiation has taken place, because both will be rich in the meristematic tissue. So, we will take this part, then we will sterilize it. Sterilize, after sterilizing what we will do? We will excise it, means we will cut it in such a pattern that it becomes of a proper size for plant tissue culture. So, generally it is small, generally the size which is preferred is around 800 micrometer maximum. So, this length of the root we will be taking. After that we will be transferring it to a fresh medium. What will happen? The lateral roots continue to grow and provide several roots, which after 7 days are used to initiate stalk or experimental cultures. So, here what will happen, that rather than having the callus, the lateral roots are going to produce more and more lateral roots itself, and we keep on culturing the lateral roots. Now, the benefit of this is that if you are having a drug where you have the roots as the main drug, like example you have licorice, that is mulethi. If licorice is your drug, then you can do its cultivation in the lab itself by directly, rather than growing the entire plant, we are directly growing the roots only. So, that our main focus or our whole energy for growth is going towards the roots and not the entire plant. So, this is the benefit of doing the root culture. The root material derived from the single radical could be multiplied and maintained in a continuous culture, and such genetically uniform root cultures are referred to as clone of isolated roots. Then leaf or leaf primordia culture. Here again we take about 800 micrometer of the leaves. They are detached from the shoots, then they are sterilized and placed on a solidified medium where they remain in the healthy condition for a long period of time. Growth rate here depends upon at which stage we have taken our leaf, whether it is a fresh leaf or a old leaf. Young leaves have more growth potential compared to the old leaves. Then the shoot tip culture. Here the excised shoot tips, which are in the range of 10 to 1000 micrometer long, are taken, and then they are basically you can say first they are disinfected, and after disinfection they are washed again, and after washing they are put into a medium where the shoot starts growing into further different type of stem parts. You have more and more shoot being cultured in the nutrient medium. You can add further growth hormones, and you can also change the different type of growth hormones which can lead to the formation of roots from the stem itself, or even you can develop the entire plant from the stem itself if we are applying proper amount of the plant growth regulators. Now, we come to the complete flower culture. Now, in flower is a very complicated part organ of a plant. Here you can use different types of parts, and it can give you different results. So, first culture was done by Niche in 1951, and he reported the successful culture of the flowers of several dicotyledonous species. He was the first person where he took the entire flower and he first of all disinfected the flower and put them in a culture medium where they started to grow into, first of all the flower won't remain as it is, it starts converting into a callus like substance, and then the callus was developed into a new plant. Now, you could develop flowers itself again by adding different growth regulators, or you can develop the entire plant from the flower itself. The flower, it remained healthy and developed normally to produce mature fruits. Flowers, they are 2 days after the pollination are generally used. You are not going to use the young unpollinated flowers because they will not be of that use, because they will only have the one part of the parent plant, the other part of the parent plant won't be there in the flower. So, for having both the parts, it is used after 2 days of pollination where the zygote formation has taken place, and then after that they are excised, means cut into small part, and then sterilized by immersion in 5% calcium hypochlorite solution. They are washed with sterile water and transferred to culture tubes containing an agar medium. So, after that they convert into a callus, and then the callus can be converted into an entire plant or a plant part. Now, from in the flower itself we know that there are different parts. So, one of the important part is the anther or the pollen culture. So, in a flower you have the anther like this is the anther, this is the filament part, and this is the anther. Anther is containing pollen grains, and the type of pollen grains present in the anther depend upon the age of the anther. What is the age of the anther? But if it is a young anther, it will have less number of pollen grains. If it is a matured anther, then it will have more number of developed pollen grains. And depending upon what is our aim, we have to select that whether at which point of time we have to select our anther or pollen grain. So, here young flower buds, they are removed from the plant and again surface sterilized. Surface sterilized means you are just applying any kind of hypochlorite solution. Sometimes you can use your ethanol also, 60% or 70% ethanol can be used for surface sterilization. The anthers are then carefully excised and transferred to an appropriate nutrient medium. Anther at very young stage containing microspore, mother cells or tetrads, and at late stage means containing binucleate starch filled pollen of development are generally ineffective, and hence for better response always select mature anther or pollens. So, we should not use those anthers or pollen grains which are immature or over mature, because they will not give us good results. We have to have proper mature as well as active form of the pollen grains and anthers, which will prove to be more effective in producing the good quality plants from the plant tissue cultures. After this comes ovule and embryo culture. Now, in a flower, the ovule is the area where the ovule, if it is an unfertilized form, then you will have only one part of the cell. And in the ovule itself, if the pollination takes place, it gets converted to a embryo having the properties of both the male and the female plants. Okay. So, embryo, it is dissected out from the ovule and put in the culture medium. Very small globular embryos require a delicate balance of the hormones. You have to be very you can say you have to take care while you are adding the different plant growth regulators or the nutrients to this embryo, because embryo size itself is very small. Secondly, it is very you can say it can be broken or it can be lost due to its size inside the culture. So, we have to be very careful when we are performing the embryonic culture. The mature embryos, they are generally excised from the ripened seeds. The other pattern can be, rather than taking the flower and then excising the embryo from the flower ovule, we can wait until the seeds start developing. And when the seed is ripened, at that time if we excise the embryo, then the possibility that it will survive for a longer period of time, it increases. So, rather than taking the embryo from the flower, why not to take it from the seed, so that the viability of the plant keeps on going on. So, matured embryos, they are excised from the ripened seeds and cultured mainly to avoid inhibition in the seed for germination. This type of culture is relatively easy as the embryos require a simple nutrient medium containing mineral salts, sugar and agar for growth and development. So, the type of the nutrient medium is also very simple if we are using the embryo from the seed culture. It is preferred than the ovule culture or the embryo from the ovary part if we are extracting and we are using it for doing the culturing. And now we come to the important type of culture, which is the protoplast culture. Now, what is a protoplast? In a normal cell of a plant, you have, this is the nucleus and you have two layers of cell covering, then you have many types of organelles which are present. Okay. So, the outermost layer is known as the cell wall, inside it is the cell membrane. So, when the cell wall is removed, still the cell membrane has the capacity to multiply and produce newer cells. So, the activity of the cell does not get affected if the cell wall is removed, and this cell wall is composed of cellulose. Okay. Cellulose is a type of a carbohydrate and it can be easily degraded by the enzyme named cellulase or macerozyme. So, in this type of culture, what is done that the plant cells, they are treated with the enzymes cellulase and macerozyme, which degrade the cell wall. Once the cell wall is degraded, the cells become, you can say, they are free of the cell wall and only a cell membrane is there on the outer side, and this particular part is known as a protoplast. Any kind of procedure done in a lab or biotechnological changes, or you want to make any kind of variations in the cell, that is possible in the protoplast compared to a normal cell. The cell wall makes the things very difficult at times for the biotechnologists to perform different types of studies. So, it can be done by removing the cell wall. So, here protoplasts are the naked cells of varied origin without cell walls, which are cultivated in a liquid or a solid medium. You can cultivate it either in a liquid or in a solid medium. Protoclast can be used by isolated by mechanical or enzymatic method. The better method is the enzymatic method. However, by mechanically also we can crush out the cell wall and remove it and make our protoplast completely naked. Okay. So, we can use either mechanical or enzymatic method for all the parts of the plants, like we can use the roots, tubers, root nodules, leaves, fruits, endosperms, crown gall tissues, pollen mother cells and the cells of the callus tissue as well. So, we can use all these different parts which we have been performing in the plant tissue culture, and using the techniques either by the mechanical pressure or by enzymatic methods, by using cellulase or mesozyme, we can convert the normal plants into a particular plant cell into a protoplast. So, I will further take you to this diagram where you can understand the whole process of protoplast culture. We have taken a leaf from this particular plant. This is a plant where we have taken the leaf. We have first done the sterilization of the leaf. After sterilization, what we did, that we peeled out a epidermal layer from the leaf. After peeling, this is how it looks like. The peeled leaf segment looks like this. This is the epidermal layer. After that, we plasmolyze the cell in an enzyme mixture. We added mesozyme or cellulase to this particular plant cells. Sometimes both are also used. Rather than using only one, we can use both the enzymes. After that, we could see that the wall digestion started, the partial wall digestion started. After this, this whole mixture was centrifuged at a very high speed, so that the cell wall proteins, they settle down, while the protoplast, they remain inside the upper layer liquid medium, dispersed in the liquid medium. So, after centrifugation, whatever the particular protoplast were obtained, these were isolated from here. They were then plated into a culture medium. Further, they were treated with nitrogen nitrate and sodium nitrate, and after that we could perform the fusion of the protoplast. Further, after fusion of the protoplast, we added certain external chemicals which started the regeneration of the cell wall. So, the wall regeneration took place. After wall regeneration, normal cell division took place, normal cell clumps formed, a colony formed, callus was formed, callus was differentiated, a plantlet was formed, and from the plantlet the entire plant got regenerated. So, the main step here involved is that we have used the mesozyme or cellulase for crushing out or removing the plant cell wall from the plant cells, and after that the next step which was important is the centrifugation, where the cell walls got separated and a pellet of protoplast was formed. This pellet was then plated into a particular medium where we fused it with other protoplast and developed our new type of cell, and after that a callus was formed. From the callus we could regenerate our entire plantlet. So, here the fully expanded young leaves from the healthy plants are collected, washed with running tap water and sterilized by dipping in disinfectant for about a minute, and then they are treated with 2% solution of sodium hypochlorite for 20 to 30 minutes. They are washed again with sterile distilled water and make it free from the hypochlorite solution. The lower surface of the sterilized leaf is peeled off, and then the stripped leaves are cut into small pieces, mainly the midrib area. The peeled leaf is segments, they are treated with enzymes like mesozyme and then treated with cellulose to isolate the protoplast. The protoplasts so obtained are cleaned by centrifugation and decantation method. Finally, the protoplast solution of known density, that is 1 into 10 raised to the power 5 protoplast per ml is poured on the sterile and cooled down molten nutrient medium in Petri dishes. So, the plating is done. Now, we generally mix this content, means the medium as the protoplast mixture, they both are mixed nicely, gently rotated, and then we allow it to set, and finally we seal the Petri dishes with paraffin film. Incubation of the Petri dishes is done in the inverted position in BOD incubator, and the protoplast which are capable for dividing undergo cell divisions and form the callus within 2 to 3 weeks. The callus is then further subcultured on the fresh medium, and from this callus embryogenesis begins, and it is then this new embryo, it is transferred into a proper medium where you have the auxins and the cytokinins, and these these plantlets are formed from the callus which is transferred into a new medium. So, with this we end the few types of plant tissue culture. In the next lecture, we will be continuing further with the hairy root culture as well as the suspension culture. Thank you.